So microbiological assay of vitamin B12. Vitamin B12 is also called cyanocobalamin. We can find out the concentration of vitamin B12 by using a microorganism. In antibiotic assay, we have seen that the antibiotics can inhibit the microorganism, and we try and measure the inhibition. Here, vitamin B12 actually helps in growth of the organism. So here we try and assay the B12, trying to find out how much growth it supports. And this concentration of B12 is compared with that of a standard. So similarly the method is done. You have an organism, you have a medium, and to that particular concentration of vitamin B12 is added, and we try and measure the growth. So first again you need an organism. For vitamin B12 you have a definite organism which is used. It is called Lactobacillus leishmani. Lactobacillus leishmani. Now this organism it grows well in presence of vitamin B12. So B12 is required for the growth of this particular organism. So we try and measure the growth, enhancement of growth by vitamin B12 of this organism. Then you need a medium. And then you need the concentration of vitamin B12. So different concentration as per the pharmacopoeia, the concentration is prepared, that of the sample and the standard. So you have sample vitamin B12 and standard vitamin B12. The samples are prepared. Serially they are added into the tube along with the medium. Medium is also selected. You have a specific medium which is used. Like similarly for like vitamin B12, we can do it for even amino acids. So accordingly you have the medium prepared such that B12 is required for the growth. If B12 is not there, the organism may not grow. So we take a positive and negative control, and then the test is done. So what is the test? The test is medium with the different concentrations of the B12 are treated with the same amount of organism. So there is inoculation of the organism. In the medium we add the vitamin and the organism, and we incubate it for a particular period of time. Different concentrations can be taken. We can have a statistics which can be applied, and the incubation is done. After incubation we measure the growth. Either it is the turbidity of the growth is measured, or we can do the growth is measured by turbidimetric method, or it can be the growth can be titrated with NaOH, and we can try and find out what is the concentration of NaOH required to neutralize the acids which are produced in that, and it can be compared with the standard. So one method is different standards, different concentrations can be taken, a graph can be plotted, and the sample's concentration can be found out by looking at the sodium hydroxide titration titer value or the growth. Or another method could be like how we do factorial assays. The lowest concentration, the highest concentration which supports the growth can be taken, and it can be compared with the standard, and we'll have an assay of the vitamin concentration required to promote the growth of the organism. So similarly we can do the assay for other enhancing agents. Inhibiting agents is separate and enhancing agent is separate. So cyanocobalamin can be assayed by this method using lactobacillus. By measuring the microbiological growth, we can assay it and compare it with the standard and determine its potency.